doing a research project, and it's very exciting. We're collecting data that's going to be used to find biomarkers for early detection, to help with quality of life issues, and a number of other things. So if you could participate, it only really requires about 10 minutes per week. Our goal is 1,000 participants. We need people who are diagnosed with ALS and people who are not diagnosed with ALS. And if you feel that you can't participate, we would ask that maybe you reach out to any of your friends and family, because it'll really help us move forward and push the needle. We're looking at this in a really different way, and we're making the data available to many, many researchers, which is a little different way. And I think that's going to make real progress for us. So thank you very much. You can just go to our website, click the research button, or you could always email me from the website.
And foremost, say thank you to our honored guest, Dr. Robert Brown, comes to us as the program director in neurotherapeutics at the University of Massachusetts Medical School. He has been, like he said earlier, working in this disease process for many, many years, and we are blessed and honored to make his acquaintance and listen to him speak tonight about genetic therapies in ALS. So without further ado, Dr. Brown.
Thank you. So I'll again share. Terrific. So thank you so much for the chance to join you tonight. I'm going to provide an overview of where we stand in terms of understanding ALS through genetics, probably reproducing a little bit some of the very nice comments that Matthew Harms made, and then talk about where we are in terms of suppressing ALS gene. So a few words first about genetics and what I would call the roadmap toward therapy, and then some comments on actually 3 areas now of gene suppression therapy. My work reflects the efforts of a large number of people. I'll try to acknowledge them as I go, starting with the genetics in the 80s, then making models, then using either antisense oligonucleotides or microRNA with AAV to suppress ALS genes. I hardly need to say, and everyone knows this, that the fundamental aspect of this disease is the rather rapid degeneration of motor neurons in the brain, brainstem, and the spinal cord, leading, of course, to muscle denervation and progressive paralysis. What's so telling, as we all know too well, is a relentless rate of progression and the short survival. Some cases overlap with frontotemporal dementia. So the work that I do is predicated on the observations in 1880 by William Osler that there are familial cases of ALS. You'll see on the right a family tree of a man named Samuel Farr, who was evaluated by Dr. Osler, and whose great-great-grandchildren we've had the privilege of following. So these are folks who turn out to have an A4V mutation in SOD1. 3 of the 5 that you see depicted at the bottom here, unfortunately, developed ALS before the age of 40, and because it's A4V, had very, very short survival. So this defines both the challenge and the opportunity, and that's where we started. The obvious perspective, which I'm sure you all know, is that, of course, if you can find ALS genes, and maybe even there are genes or toxins in sporadic ALS, but if you know what the upstream trigger is, then you can maybe figure out the steps downstream that lead to the disease. So our goal all along has been to try to define these arrows so that we could potentially define targets. And we presume that, yes, of course, in the familial cases, it's mainly genetic disease, but we all still, I think, believe at some level environment and potentially neurotoxins play a role. So in the early 1980s, we undertook to look for ALS genes. This is the first family that we studied, fALS 001. You can see in black the affected individuals over several generations. We studied it with a large and wonderful group of investigators in Boston. Jim Gazzella's lab started it. I picked it up after that. Bob Horvitz at MIT was absolutely essential. And at the same time, a large group of very good investigators at Duke undertook the same. Important is that now there is an army of people looking at ALS genetics, and I've depicted several of them here. Many of these are very well-known neuroscientists, and I'm sure you know of their work. Just as a quick aside, it has been really quite an odyssey looking for the genes. We found SOD1 after collaborating with a huge family in Belgium, in Leuven or Louvain, depending whether you're Flemish or French, with Wim Roberek. So this was a whole new encounter. We were very lucky to find the FUS gene from Cape Verde, with a very small family, but one that turned out to be very instructive. We found a very interesting gene called profilin-1 from an Israeli-French family. And this required, I'm delighted to say, travel to these areas to see these folks acquire their DNA and do the study. So it's been a long quest, but it's had a wonderful upside. But here's the real upside. As you know, there are now scores of ALS genes. This shows the number as a function of time. And the amazing thing is that, you know, depending on how you count them, there are 20 or 40 or 60 or more, again, depending on how robustly you take the data. In the United States, the most abundant is C9 ORF72. They hardly see that in the East. In fact, their SOD1 is the most common ALS gene. Others follow suit. And I'll tell you now that we probably understand about 80% of the genetics when a familial ALS case comes to us, and even probably 15% of sporadic ALS. So one of our themes was from the very start that if we understood familial ALS, it would tell us about sporadic ALS, which of course is 90% of cases. So that is one of the themes I'll come back to later. Well, one of the immediate things that was quite wonderful in finding these genes is even if you don't understand them, you can take the gene and put it into a mouse, and you can, incredibly enough, generate a mouse model. So here's a mouse model of SOD1 ALS. These animals are perfectly fine until they're, 110, 120 days old, and then they rapidly develop first hind limb, then forelimb paralysis. Here's an animal which has the profilin-1 gene put into it, and it too develops paralysis. And as I'm sure you know, what's also very, very exciting is that it is possible now to recreate motor neurons in a petri dish from the patients you see in the clinic using stem cells. So you take a skin cell or a blood cell, you make a stem cell, and you make a motor neuron, and voila, now you have a model of a patient's own motor neurons in a petri dish, which is pretty amazing. Well, what do these things do finally to the nerve cell? Well, this is meant to be a cartoon of a motor neuron, and what this is meant to show you is the cell body in yellow, the axon that it sprouts to go down to muscle, shown in brown on the right. And I think the main point to say is that almost every part of the motor neuron gets sick in this disease. So there's unplugging, as you know, of the distal terminal from muscle. The way the terminals transport materials up and down is abnormal. The firing patterns are abnormal, with excessive firing in at least part of the disease. And a number of other things are abnormal, such as the manufacture of special RNAs that control RNA development. Mitochondria, which make energy, are defective in many cases. And protein quality control is defective as well. And the interesting thing that makes this so challenging is that different parts of the motor neuron will probably get sick at different times. And what this tells us, I think, is that to treat this disease, you're going to have to treat more than one process. I don't think there'll be one pill. There will not be one drug. There will be a lot of different things that will have to be treated simultaneously, and perhaps even at different phases of the illness. So the complexity is writ large. Now, how do you relate this to the genes? Well, it looks like there are sort of three categories of those genes that I'm sure Dr. Matthew Harms would have talked about. So one has to do with a set of genes that control the quality of proteins. Do they form and function normally? Another set of genes has to do with essentially how the long process of the axon grows and maintains its stability. And the largest set, which we never expected at the start, is a set of genes that make proteins that have to do with how RNA is handled in the cell in various circumstances. But at least we have sort of three pathways to think about. Now, the other pathway to mention to you, which was not known at the start, but which clearly is present, is the following. There is a large degree of what you might call neuroinflammation. So in addition to all these things going on in the motor neuron, a lot of other cells get activated surrounding the motor neuron in a kind of inflammatory way. And inflammation in the nervous system is just like it is in the skin. It has good functions and it has bad functions. So figuring out how to modulate it in a favorable way remains a challenge. But I will tell you that there are many drugs in the pipeline now that are targeting not the motor neuron, but the neuroinflammatory process. And another really important point, which I won't spend a lot of time talking about, is that by the study of the familial genes, there have been disclosed 3 different genes that are also very, very important in non-familial ALS, in sporadic ALS. And I've listed them here just sort of cryptically: a gene called TDP43, another ATXN2, and a very important gene recently discovered is called STMN2. And the point is that we learned about these genes by studying familial ALS, but these probably pertain to all cases of ALS. And that was, of course, the kind of dream and the hope at the start of this search for genes, that we now have something that gives us a perspective on all types of the disease. So how do you treat this? Well, I'm sure Matthew would have talked about this a little bit, but quite obviously, when you look at a kind of simplistic diagram like this, you got two big choices. One, you can go way downstream and try to treat all these many processes going on in the motor neuron, or maybe you can go upstream and try to treat the trigger, try to treat, if it's a familial case, the abnormal gene. So I'll just tell you that there are now many, many downstream therapies that are either currently in trial or soon to go into trial for ALS. I keep a running chart, and I think, no joke, I have about 70 drugs at various stages of the pipelines, and most of which target these things downstream. They target things like mitochondria, cell stress. They target things like the level of this gene ATXN2, modifying either TDP43 or STMN2. And part of the reason why I think we're all very upbeat about where we're going in therapy is that the downstream treatments do look promising. So let me just mention a couple of them. So I think I saw the name of Dr. Nathan Staff on the call. We were, well, I'll get to that in just a minute. I'll jump around it. We were privileged to work with our colleagues at Mayo Clinic, including Nathan, to study stem cells and how they might benefit ALS. And the jury's kind of not entirely in on that, with some wonderful and notable exceptions. But here's what we learned, that when one puts these stem cells into the spinal fluid, there clearly are some benefits. One is that there are a number of inflammatory factors which go down. So here in the bottom, you see patients who didn't receive the stem cells, and in the top row, people who did. And in black is pre-treatment and brown is after treatment. So here are two inflammatory factors that went down significantly after stem cells were given. And then analogously, if you look at this panel on the left, again, pre-treatment black, post-treatment brown, what you see is an increase in three factors which are very important in sustaining neuronal viability. So this is one way to go downstream. And I can tell you that, as you can see, these multiple factors will have multiple effects downstream. It's not just treating one effect. The other super exciting data point that came out this summer was this finding, and I know you've all heard about this, that the combination of two drugs, sodium phenyl butyrate and taurursodiol, are beneficial in ALS. They have different functions. They're downstream again, but two papers came out, one in the New England Journal, that's pretty darn good, which showed slowing of the rate of progression with this combination, and a second paper shortly thereafter, increasing survival. So here are a couple of examples of downstream therapies which are important and need to be taken forward. But what about the total field? Well, we can go over this later. Again, this is a sort of short list of what's out there in the pipeline now, but it's very exciting. So how about the upstream therapies? Well, let me comment on that. So the central dogma is, you know, I'm sure, and the whole concept of how genes work is that the gene is made up of DNA, and it works through a template, which is almost the same chemistry, but not quite the same, called RNA. The DNA generally stays in the nucleus. The RNA is in the nucleus, but can go out into the cell. And out in the cell, it makes protein. So this is kind of the set of pathways. And so if you think about going way, way upstream to treat the causes of ALS, how can you intervene in this set or sequence of events? So, well, there are ways, for example, to edit the genes. We've all read a lot about it. This so-called CRISPR-Cas9 is a way to go back right into the DNA, correct the mutation. And in a way, that's the holy grail. Now, there are some diseases where the problem is that the gene is missing. And in that case, for example, you can use a virus and a cargo, which is the missing gene, but now the normal version, to go in and replace the gene. So that's really upstream. That's treating the DNA one way or another. Another thing, of course, you can do is say, hey, this RNA is making something poisonous. Let's kill the RNA. And you can do that with antisense oligonucleotides and other approaches such as microRNA. And then finally, if you say, gosh, the real problem is the protein, can I treat the protein itself? Well, there are ways to deplete toxic proteins. And, you know, Biogen, for example, has tried antibodies to this protein that's involved beta amyloid in Alzheimer's disease, and indeed strategies like that have been looked at in motor neuron disease. Does gene replacement work? Well, this is a side road, but in fact it does. This is one of the most extraordinary stories in our time, and it gives us hope for ALS. So this is a little baby who has spinal muscular atrophy. And these are the saddest case you've ever seen. Beautiful little baby, very smart, very bright, but very weak, because this baby has motor neuron disease. It started in utero, and it's so severe now that this poor little baby, barely 3 months old, cannot breathe, can't hold the head up, the head lags. This is just a heartbreaker of a disease. And look at this beautiful little baby. And look at the eye contact. You just want to reach out and hug this little child. Well, it turns out that gene replacement therapies and gene modification therapies have made a profound difference in this disease. So it now is no longer as lethal as it used to be, because there are two different beneficial approaches. So my point is that these interventions at the genetic level are no longer just pie in the sky. They're real and they're with us. So coming back now to ALS, we have two genes that have quite appropriately deserve the most attention. There will be many more to follow, SOD1 and C9 ORF72. And for each of these, again, go back to that canonical model. You can theoretically use DNA targeting with CRISPR to edit the gene, make it right. Or you can go after the RNA, and that's been done already with antisense oligonucleotides, or ASOs, as we call them, and microRNA. Or you can try to deplete the protein, the mutant protein. And Jean-Pierre Julien at Laval and colleagues in Japan have done that with some promising results in mice. Or another approach is to come up with small molecules that shut down the whole network that leads to the production of these genes. This is not something that's being pursued too much. We did that with ALS. We found a small drug that looked promising. Tipu Siddiq at Northwestern has the same. There's an incredible man who used to be a head of NIH named Harold Varmus, who's a Nobel laureate, who was studying cancer. And he found a small molecule that turned off a pathway that made cancer cells grow. And that pathway that was turned off happened to be SOD1. So no less a person than Harold Varmus discovered a drug that can turn off SOD1. So these are all a set of choices that are there. So let me just give you some very specific sort of examples. So the idea that these antisense oligonucleotides can shut off SOD1 has been around quite a while. We were privileged shortly after SOD1 was found in 1993 to work with Jeff Rothstein and show that we could turn off SOD1 with antisense oligonucleotides. And a couple of years later, Michelle Maxwell in our labs showed she could turn off mutant SOD1. So feasibility was clear from the beginning. Beautiful work done by Richard Smith, Don Cleveland, Tim Miller showed that in a rat, they could make antisense oligos that shut down or essentially the gene 50–60% and prolong survival. And this led Biogen, working with Ionis, to say, let's see if we can make a commercial product out of that. And that led to the first trial of ASOs to suppress SOD1, actually now 4 or 5 years ago. Low dose, but did prove safety. But the real exciting news, which I know you've all heard, which came out in the New England Journal this summer, is that the lead ASO from Biogen actually shows fairly considerable efficacy in shutting down SOD1. It prolongs the life of SOD mice. But what they showed was that at least in a dose-dependent manner, it reduces the level of SOD1 in the spinal fluid. And in a small group, admittedly, a small group of rapidly progressing patients, it actually slowed the rate of progression. So the treated patients here are in brown, and the untreated patients are in blue. It's a very big difference. Small numbers, needs to be repeated, but very, very exciting and encouraging work. So our own approach has been, we actually have an ASO project, which is a whole other topic, but to look at microRNAs, which are slightly different and which have to be given via a virus. The strength, if it works, of a microRNA approach is, because it's given by a virus, in theory, the virus, once in the cell, will keep making the microRNA. So this could be one treatment lasting a very long time. The ASOs, which do look very promising, have to be given probably several times a year. So we've been studying this, and I won't bore you too much, except to say that shown in the bottom here is the survival curve of our ALS mice. I showed you a picture of an SOD mouse. And in blue, you see that all of them are alive at about 110, 120 days. By 150 days, they all are dead. But when we treat them with one shot of the virus with this anti-SOD substance, they live a long time. As you can see here, 206 days. So this mouse shown it right is not normal. Look, it's got a curved back, but it can crawl, it can rear, and it can crawl upside down. That's a pretty darn strong mouse. It has just as much toxic SOD in the genetic component as the untreated mice, but its SOD has been silenced by the microRNA. So we then took this approach and we tried these microRNAs in normal monkeys, 2 or 3 different times, different strains, and the microRNA seemed to be safe. So we've then gone on to do a trial now in 2 patients. I'll just go through this briefly, but this is work done with our colleagues at the Mass General, Merit Sokovich, James Berry, Chris Mueller, then at UMass, and then our wonderful team, the trials team, Kat Douthright, Dr. Wege, and Diane McKenna, in which, in the first case, we took a 21-year-old who's probably related to the same family that I showed you earlier with the A4V mutation, who came to us with very rapidly progressive left leg and foot weakness. And he knew to worry about ALS because so sadly his mother had died 10 years earlier of the same mutation. So at this point, we'd done the monkey work. We went to the FDA and we said, gosh, can we try this therapy in this young man? Because frankly, he's got a rapidly progressive course. So we used a virus called AAV-RH10. The details don't really matter. But packed in it were these 2 brown things, which is the microRNA targeting silencing of SOD1. And the thing that drives all this happens to be what's called a promoter, chicken beta actin promoter. So that's the CB here. And I'm not going to spend a lot of time on this, but I'll just point out a couple of things. So the bad news part is his disease was pretty well rip-roaring, and it kept going. So by the time we got to him, we were concerned that we might be closing the barn door after the cow has fled, and that could indeed be the case. And so if you look, for example, at grip strength, what you see is relative strength in the y-axis at the top here over time. Now this is actually, it says knee flexion extension, but they're down below. This is grip strength. And what you can see is that the left side, which had been slightly affected, really continued to progress rapidly. And the right side, which we thought was pretty normal at the start, also went very, very quickly. So we didn't help that at all. The left leg, which was what brought him to attention, was so far gone that we could hardly measure anything, as shown here, looking at knee extension and knee flexion. But what was interesting was that the right leg showed some continued progression, but then it seemed to stabilize. And I can tell you that 14 months later, he was still able to push himself around in a wheelchair with the right leg, suggesting that in some way, this rapid trajectory had been improved by this intervention. Now, this is N=1, so I acknowledge that. But we've not seen this in A4V SOD ALS before. So just briefly here, what also interested me was this, that 21 weeks after he'd been treated, he called me from his home. And he said, look, doc, I'm going to send you a video. I've recovered some ability to move my foot, which he hadn't done for several months, which gave us some reason to be hopeful. 14 months later, he said, I want you to see I've recovered some function in my left hand, which was also not something we've seen before. So these points at least gave us some hope that this was a beneficial therapy. Now, what else I think was encouraging is as follows. When we looked at the motor neurons in the cervical, that's obviously the next spinal cord, and when we look at this is the whole spinal cord, and the portion at the bottom here that is in the dotted box is where the motor neurons are. And I'll just tell you, whether you look on the left or the right, you don't see very many because they're pretty well gone. And that correlates with the hands going. But what intrigued us was that on the right side in the lumbosacral spinal cord, where the motor neurons or the legs are, there was remarkably good preservation of motor neurons on the same side where he had preserved strength. Again, suggesting that for some reason, maybe it was chance, maybe he's 1 in 1,000 by chance alone, or maybe it was the benefit of the therapy, there was some benefit. And this, of course, has propelled us to look further. Also, I won't bore you with the fine print here, but I'll just tell you, when we looked at the level of the protein of SOD1 in the spinal cord, both in the base of the cord, the lumbosacral cord, and in the neck, a band here looking like light gray is the level in the patient. The level in 5 other SOD A4V patients, it's now renamed A5V, is shown here in lanes 2, 3, 4, 5, and 6. A huge, huge difference. This was really substantially depressed, and these are healthy controls. So this was another data point that said, maybe this has had some benefit. Now, there was a downside. This is the first time this has ever been used in a patient at all, and certainly in ALS. And a downside was that when we gave the virus, he developed a lot of inflammation to it, okay? And that was manifested in a couple of different ways. One was that he had pain. He had a pain syndrome, which came from the inflammatory process irritating his sensory nerve roots. He also had some cells in his spinal fluid, and some of this spilled out into the bloodstream, and he had some elevation in markers of cellular activity in the liver. So without going into too much detail, what you see at the bottom are 2 inflammation indices over time. And they both went up by about 6 to 10 weeks and then slowly kind of subsided. So we thought, given all this together, that we'd like to do another case and maybe try to find a way to treat the inflammation. So what you see in the second case, where we used the same amount of virus, it was a different mutation, which was not as rapidly acting. We were able to suppress the inflammation. So in case 2, you just don't see the inflammation that was there in case 1. So what we concluded from these 2 cases was this could be tolerated safely, particularly if we suppress inflammation prospectively. Secondly, there seemed to be a real evidence of suppression of the protein in the spinal cord, and there seemed to be a hint of clinical benefit. So we're very anxious to try to take this forward. So let me switch gears now a little bit and move to the C9 gene. So we've worked very, very hard on this now for about 3 years. And I won't spend a lot of time talking about the molecular biology. Most of you, I'm sure, have read this, but this is the gene here. And the gene has these working bits, which are in blue, the exons, and then they are arrayed on a backbone of DNA, which are called introns in gray here. And the defect in this most common type of familial ALS is that there's a segment up in the beginning of the gene that has 6 nucleic acids, G, 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 C, C, which normally occur in, say, 3, 4, 5 in a row. Some of us might have 12, 15. But in the patients with C9 ORF ALS, they have not only hundreds, many of them have thousands of these. So this piece of DNA just expands enormously. And it does a number of things. That helps really suppress the gene so it doesn't make enough of its normal product. But it also leads to the production of some proteins which are very toxic. And for the moment, if you'll allow me, I'll just use their alphabetical names: GR, GA, GP, PR, and PA. And so this is part of the pathology. So our interest was the following. It turns out, remember the canonical gene saga. You got DNA, it makes RNA, it makes protein. And it turns out that when you look at the RNA that's made from C9orf72, it makes 3 different RNAs. They're called variant 1, 2, and 3. And V1 and V3 are the ones that have the expansion. So our goal was to make antisense oligonucleotides that would target this expansion, but spare V2. And most of the protein is made off of V2. So the dream was that if we did this, we could shut down the offending parts of the gene, but leave a functional part intact. And this is work done by Jonathan Watts at UMass, who's a fantastic nucleic acid chemist and really bioengineer. Mike Mozami, great work by a postdoc, Elaine Tran, and Courtney Pinto. Really great team. We were blessed to have them. So bottom line is, our goal was to shut off V1 and V3 and leave V2 intact. And I won't go too far into the weeds here, but I will tell you that we looked at a large number of antisense oligonucleotides, and we did find one that seemed to turn off. This is V1 and V3. This is higher and higher doses, showing we can shut it down. But as those doses increase, V2 is more or less constant. And we could show that this effect lasted for 20 weeks. We also could show that that toxic protein, poly-GP, was also turned off in a dose-dependent manner. And that effect lasted also 20 weeks. So we tried this in a series of sheep just for safety purposes. It was indeed safe. And then we tried it in a series of monkeys at varying doses, and it was safe in monkeys. So the FDA gave us permission to try this in one human, and we've now treated this one person for, I think, just over a year. He's got a well-defined family history, I'm sorry to say, and a clear expansion. And what we found is that he, like the sheep and the monkeys, has tolerated this pretty well. And it looks like there's benefit in terms of reduction of the GP levels. And then I'll end with just one other story, which you may have heard about, and that involves the gene called FUS, F-U-S. So in a word, it turns out that there's a small set of kids, really, children, age 12, 14, 16, who get really rapid motor neuron disease. And in the past, it was so rapid and so inflammatory that people didn't even call it ALS. They called it, myeloneuritis or something like that. But it turns out when we found the SOD gene and started to look at those cases, those are FUS, sorry, the FUS gene, those are FUS cases. And so that's another gene that's super important because some of its mutations cause really rapid disease in really painfully young kids. So I saw, had a chance to study the DNA in 2006 from a 14-year-old who had rapidly progressive ALS. And she was thought to have this inflammatory disease, but when they looked in the spinal cord, she had abnormal FUS. And when we studied her DNA, she had a very bad mutation. And the thing is, she had a twin sister, an identical twin sister, who didn't have the disease. And so you knew that that identical twin was at high risk. So basically what happened in this story is that about a year and a half ago, almost 2 years ago now, this sister, whose name was JC, had developed ALS, and it was starting to move very, very quickly. So we were very fortunate that Frank Bennett and Payman Jafarnejad at Ionis allowed us to study an ASO that they had that targeted part of this FUS gene. Again, we studied this with Jonathan Watts and two other wonderful people in my lab, and then shared it with Neil Schneider, who was taking care of JC. And the bottom line is that after this looked like it might have some benefit in FUS mice produced by Don Cleveland and Sandrine de la Cruz, Neil Schneider treated JC with this. And, I can only say that it was safe, and it may have had a transient benefit, but unfortunately, she was so far advanced by the time the treatment was started that it really didn't have an opportunity to show benefit. But what I'm really trying to say is that there are examples right there. Essentially, SOD, microRNA, ASOs, the work with C9, and what's wonderful is that Biogen Ionis has got a great program treating C9 with ASOs also, the FUS work, and there are some others now in progress that all suggest that there will be what you would call personalized medicine approaches to treat these inherited forms of ALS. Now, what's particularly exciting, to come back to that bridge between sporadic and familial ALS, is that those three genes, TDP43, ATXN2, STMN2, also provide targets for therapy, which we think will benefit sporadic ALS. So we and many other labs are working very hard on that particular new flavor of investigation, building on these early stories. So what I conclude is all written here. It's extraordinary. We didn't know at the start if there'd be any ALS genes. Now there are scores of them. That's amazing. I think they do tell us about sporadic ALS, which is what we hoped in the start. The therapies, both downstream and upstream, are extremely promising. I fully, deeply believe that personalized therapies are going to really help us for targeting specific genes. And then what we've learned from these last few cases is that if there's even only one or two cases and things are just right, you can move very, very quickly to a personalized therapy for small numbers of patients in a very short time, provided that the FDA allows you to test the substances. So I'm overall very, very upbeat about the field and where it's going. And I'll stop there. I'll be very happy to take any questions.
Thank you so much, Dr. Brown. We are incredibly honored that you're here with us, and because he's gotten on the call with us to speak to some of the legislative team to help fight for expanded access. So we love you for your scientific expertise, but also because you've been an advocate for everybody in the ALS community, and that means a lot to us. So as we get started, I'm going to go back to the first question, and then Sarah and I are going to go back and forth. The first question we have for you goes back to the virus treatment. So what's the current status of the SOD1 suppression with the AAV, with that virus and microRNA? Where is that research right now?
Yeah, so a couple of things. So it's underway. The fact is that, and your questions later on allude to this. So UMass spun out a company called APB102 to take this forward. I mean, just a quick aside, as everybody knows, this is unbelievably expensive. So we could make ASOs to try to treat a patient for $30,000 or $40,000. The virus costs about $1 million each time. That's a huge amount of money for an academic to earn. So at some point, when there's a hint of proof of concept, this really has to move either into the hands of folks who have very large resources or into a company. So UMass spun out a company to take this forward. Again, it's called APB102, and they have developed a program, which I hope will start by the end of the year, to try to get this into a large number of individuals.
That's going to be a phase 2?
Sorry?
It will be a phase 2?
Yeah, that's right. Yeah, that's right. And as you all know, God bless them, Biogen and Ionis is galloping forward with their SOD1 ASOs. So this is two parallel tracks that offer, I think, real hope for SOD1 ALS. It's not as fast as we want it to be or need it to be.
The next question says, while there have not yet been symptomatic treatment of gene causing familial ALS, can you speak about what your preclinical work is showing?
Yeah, key question. I think we have to treat as early as possible. And I've given you a couple of examples where we have therapies that are very good at shutting down the genes, but if you wait too long, it may be too late. So my best guess is that we will be setting up ways to screen people's DNA, particularly if they're in a family where there's a known ALS gene, and combine that with really close screening for early disease. And there are a number of ways to define early disease so that at the very earliest start of trouble, we can start the treatments. That's a ways down the line, but that's, in my mind, that's absolutely where we have to go.
And they went and looked at the APB102 and APB105.
Yeah.
So can you distinguish those for the people that went and looked at the website?
Yeah, well, I would just say that APB102 is the main SOD program. And they have other programs in the pipeline, and I'm not in a position to talk a lot about them now. But I would just say SOD1 is a target, and that's the one that's closest to a trial, and that's APB102. They have an interest because this is such a compelling technology in looking at some other diseases beyond just ALS. But as I think every young company should do, they're focused very aggressively on APB102.
Given the risk of inflammatory response from AAV treatments and neuroinflammation being a suspected risk of increasing disease progression in ALS, in theory, would the target demographic for AAV treatments be best suited for those in the presynaptic stage?
Pre-symptomatic stage, yeah, it would. But I say that not because of inflammation, but because of the observation that we just want to get to this before the trouble has really started. You know, the kind of simple analogy is obviously if you take one match and start a house on fire and then you blow out the match, you're not going to help the burning house, you know, and of course that's a stretch, but I mean, there is a sense that once these processes get going at some level, they become self-sustaining, and it will not be just enough to turn off the gene. One will have to address so many of those other downstream pathologies. So again, yeah, so I think pre-symptomatic stage treatment is clearly where we need to go.
Bob, you mentioned earlier in your presentation that you were talking about both SOD1 and C9 silencing. How far off is that C9 therapy from going into a phase 1 or phase 2 trial?
Yeah, well, I mean, Biogen has one already. Biogen has a C9 ASO in trial. So it's happening. And I showed you the data from our one case. So these are happening and they look very promising.
So you think that that's in the next year or two, you might be able to get that C9 into the trial? Into a phase 2 trial?
They're already in trial in humans, and whether or not it'll be a large enough set of humans to really, as you say, essentially produce a trial that will lead to a registered drug with confident benefit is unclear to me, but it'll happen. It's just a question of how rapidly these can be scaled up. But it's clearly happening. It's not very far away. But anything is too far away if it's not tomorrow. So, yeah.
For those on the call that may not fully understand the mechanisms, can you kind of explain the difference between the ASOs like Biogen's and Apex genes silencing treatment and how
Yeah, that's a good question. They're really quite comparable. They both tackle the RNA. So remember in that sequence, gene, RNA, protein, they both target the RNA. But the ASO typically works by binding to the RNA and activating an enzyme, I think it's called RNase H, which chews up the RNA, because the enzyme appreciates that these two things have come together, they've bound, and it activates the enzyme. The microRNA also targets the RNA. It's about the same length, but it gets carried into a special complex which digests the RNA slightly differently than in the case with ASOs. I think the functional difference is that ASOs are delivered as ready-to-go molecules, and they're given, they're used, the body uses them for weeks and maybe even months, but then they have to be given again. The microRNAs are given typically in a virus, which then produces the microRNA. So it takes a little longer to get going, but in theory, the virus can keep making the microRNA indefinitely, or at least years. In which case, the difference would then be that the ASO has to be given, say, 2, 3, 4 times a year. The microRNA therapy given just once. So the mechanisms are not that different. Functionally, the delivery schedules are quite different.
Thank you.
Knew you were going to get a few questions about NurOwn, so this is the first one. I'm going to tee it up. Can you hypothesize about what the possibility would be if you used either an ASO or your treatment with APIC, and then silence the gene, and then follow it up with NurOwn? Is that something that you can talk a little bit about for the people on the call?
Well, I mean, as I tried to say, I think the stem cells exert a number of downstream benefits, and it's not quite clear yet. I mean, I think there is some benefit from the stem cells. There are some exceptional individuals who've had very, very, very pronounced responses. So sure, I think if one could find a way to have that multifactorial benefit in addition to silencing a gene, if that multifactorial benefit is really a real benefit, it's probably something one would want to think about. The problem with stem cells is that they exert so many influences that we really don't know what the working ingredient is, right? And so the result is that instead of purifying what comes out of the stem cells, which is the real working ingredient, the current state of the art is you give the stem cells. And the fact is that they have to be given multiple times. So, the question is, can it be made feasible to do one or more bone marrows, grow the cells in a lab, and then give them back intrathecally? And clearly, if the stem cells produce a big benefit, everyone will do it. And of course, that's what we hope will be the case. And I think they have some benefit. But will it be enough to justify bringing it forward in addition to silencing a gene? I don't think anyone knows. I don't think anyone knows. Okay. Good question. It's a very good question.
In your presentation, you spoke about in your mice trials extending that lifespan about 69 days. Just for an approximation, how does that relate to human time?
Yeah, well, that's a good question. A mouse typically lives 2 years, 2.5 years, maybe 3 years. So, a mouse that dies at 135 days is like, a young adult, and you're basically increasing survival about 50%. So let's say if you have a 40-year-old, that would be increasing survival from age 40 to age 60, which is, of course, a big effect. It's better than what we do now. We'd like to do even better than that.
A little bit about expanded access. Given the comparatively small population of familial patients with SOD1, is that around maybe 500 or so in the US? Would that be?
Yeah.
Probably close.
No, depends. I mean, if you, what is your favorite number for the total number of ALS cases in the country? Maybe it's 20,000?
30,000 or so?
Yeah, something
I think it's like more like 35 is what.
Yeah. It's hard to
living with ALS, yeah.
But roughly 10% of those are familial. So that would be 2,000 or 3,000.
So in the context of SOD1, now that we have some promising therapies out there, given that there's some really rare mutations, what are your thoughts on using expanded access to make that therapy available to everybody? And how could that impact the clinical trial data?
Yeah. Well, I mean, if a Biogen has something that looks very, very promising, if they wanted to make it available for a subpopulation in an expanded access mode, they could. And there's no doubt that that would, I think that would probably help a lot of people. I mean, there are many reasons why a company has reservations about doing that, as I know you know. One is financial. The other is what happens if a few people have particularly bad side effects. And so the company is very sensitive to being certain that a drug in trial is administered in a way that optimizes its chance of success. So right now, most of these companies pick very carefully the type of patients they want to treat. So they have inclusion, as you know, and exclusion criteria. And so that sets a kind of filter for optimizing patients who they think will respond favorably. If you broaden that and take all comers, let's say, and as a clinician, obviously at one level, I'd very much like to do that. But if you broaden the filters and you take all comers, and then there turn out to be some individuals who have adverse effects, the question is, will that taint the drug in the eyes either of the FDA or the marketplace in some way that's unfavorable for the company? I'm making no value judgments here. What I'm trying to share is what I think is, you know, the world of pharmacy's perspective on this. As a clinician, I'd love to see everybody get it. No question about that.
In your opinion, why do you believe that the SOD1 mutation appears to be responding better to the ASO therapy than maybe the FUS gene or C9?
Well, yeah, first of all, I don't know that it's any better than C9. I think C9 response is probably just as good. In FUS, certainly the couple of cases that I'm aware of that have been treated by Neil Schneider, who's just been fantastic, have been very far advanced. And so I think one reason may be that when they're really advanced, the likelihood of response is diminished. You know, ultimately, there could be something about the biology of FUS, which is different from the other genes that makes it less responsive to something like an ASO.
Okay, Bob, so we have another one about APIC SOD1 silencing therapy. And does that work on all SOD1 variants? And if not, can you hypothesize or help give us an idea why not?
Yeah, it will target most of them, right? So remember that what happens is the ASO or the microRNA targets the RNA and binds to the RNA. And if it happens to be that a given mutation is somewhere in that binding sequence, the binding may not be normal, it may not be tight, and therefore there may not be gene suppression. So the guess is that both an ASO and a microRNA should work for most mutations that are not in the targeted binding site. And so you can arrange that, God willing, you can arrange, a lot of factors go into it, but you can pick the binding site which has the least likelihood of having a known mutation.
What are your thoughts on how the community can help accelerate genetic therapies for maybe lesser known or rare mutations?
Yeah, that's also a good question. I think, you know, I think there are two factors, or maybe three. One of them is funding. These things cost money, okay, no question. And some, like microRNA and viruses, cost a lot more money than ASOs. But one of the very exciting things about the ASOs is they can be developed very quickly for relatively less money. So one is money nonetheless, regardless whether it's a lot or a little. That really helps. The other is at some point finding a way to, and it's happening. I mean, the FDA is evolving in how it assesses these things, but somehow there has to be built in more flexibility in the way personalized medicine, particularly for severe diseases where N equals 1 or 2 or 3, which are orphanoid, where the regulatory system is more flexible at letting these treatments go forward in otherwise untreatable lethal diseases. So the issue there is lobbying, and it's not so much the FDA is our enemy and we got to shoot them down. It is they're evolving. I mean, I'm old enough to remember there's a drug called thalidomide, which maybe the old people in the room will know something about, but that was given for pregnancy morning sickness in the early 50s in Europe. And right. It was a terrible teratogen. And when I lived in England for 2 or 3 years, and I happened to be there at a time in the early 70s when all the lawsuits came due over that, and there were hundreds of little kids who were born with no arms, no legs, Didn't happen in the US. And the reason it didn't happen in the US, because one woman in the FDA said, this smells bad to me. I think this is going to cause fetal malformation. We're not going to allow it in the US. And it didn't happen. So, look, that's an extreme example, but it is a good example of where the conservatism can ultimately pay out in a meaningful way for public benefit. So the issue is, I think, encouraging flexibility and responsiveness in the FDA in a way that recognizes the direness of some of these diseases and the potential benefit of rapidly developed personalized therapies. So the community can help with that.
Absolutely. And that's a very good point about the FDA. I mean, I think that that's something that is kind of can be a little bit of a conflict, especially in this community, for the timing. And absolutely, we understand that side is just as important. So it's really working together to understand both sides and make that collaboration happen. Yeah. In terms of sporadic ALS, I know you talked about in your presentation that you think that finding and understanding familial ALS and its mutations would provide some insight in treatments for sporadic ALS. What kind of thoughts do you have behind that as to maybe why or what we could look for in the future?
Absolutely. So the ones that jump to mind are this thing called ATXN2 and STMN2. And interesting enough, both of those can be benefited by ASO therapies. And right now, Ionis is working on them. God bless them. They're working on new therapies. And I think partnering with Biogen, which is a fantastic company, to find ways to get these ASOs to regulate the level or correct problems with those two proteins, which almost surely will help sporadic ALS. Now, the question is, again, will that be enough, or because that's upstream, or do you then have to overlay these many, many kinds of therapies for the downstream management of what's already going awry in the complex cell called the motor neuron?
So with the virus insert technology, does it have an impact on the immune system?
With which, please?
The virus insert technology, that AAV technology, how does that impact your existing immune system?
Yeah, I think what happens is the virus, comes in as a foreign protein, and I think the immune system says this is a foreign protein and reacts to it and generates an inflammatory response, which can be quite significant. So it clearly does activate the immune system, much like any virus. I mean, even COVID, which we all think so much about these days, activates the immune system. That's why, by the way, we're getting vaccinated, is to activate in advance our immune system so it will attack the virus before it gets to take off. But that's essentially, I think, what happens with AAV. It activates the immune system, and then the sort of innocent bystander are the cells and the layers around the virus, which then get inflamed. And depending on how severe it is, that's quite a big problem. But we think, based on our experience, and I can tell you that UMass is treating, our dean is treating some babies with Tay-Sachs disease, which is a horrible disease. And he's putting back the missing genes, which is incredible. And now he's using the same idea of dampening down the immune system before treating these babies. And the idea is that if you dampen down the immune system, the body may tolerate the virus long enough to see the benefit. If you don't, the virus may destroy, the immune system may destroy the virus before it exerts its benefit. So your question is very important. The immune system activation can itself be very problematic.
Well, you spoke about the COVID vaccine, and I'm seeing a lot of questions in our chat specifically. Is there any reason on any of these clinical trials that are currently going on that people should not receive the COVID vaccine if they are actively in a trial?
Yeah. Well, what I would say, first of all, is that has to be addressed with each trial, with the medical management of each trial. My general view is it's probably okay. I want to just make one side comment, which is that, a lot of folks, when they get the second vaccine, get a lot of side effects. And if you're on a drug and you have, let's say, a bad 3 or 4 days of a fever and muscle aches, that then the question will then come up, is that the drug or is it the vaccine? Well, it's probably the vaccine, but I can imagine a scenario in which the side effects to the vaccine might confound understanding the drug. But each case has to be considered on its own. And in general, my own view, until I see data to the contrary, is it's probably very smart to get vaccinated.
actually brought up TEDCA and sodium phenyl butyrate earlier,
Yeah.
and some of your patients have told us that you've been a miracle worker to get SPB covered. Can you give us any insight into how you've been able to do that so they can share
No.
it with their own neurologist?
No insight at all. We write letters and tell these people that we think it works. The insurance should cover it. I will tell you, and then you just duke it out, and then you have a phone call, and you duke it out on the phone call. What I can tell you is that process is easier now because there's a New England Journal of Medicine, the premier medical journal in the country, which says this works. That makes my life a lot easier. I send them a copy of the article. I mean, how can you argue with that? Right? And so I think that's added a little clout to the argument.
So for everybody on the call, we'll make sure we send out a copy of a link of that so you can give it to your local neurologist if they don't have a lot of expertise in ALS to help you with that.
Yeah.
Wonderful. Are you able to discuss why some families affected by ALS appear to have a higher incidence of other neurodegenerative diseases, maybe like Parkinson's, MS, Alzheimer's, or SMA?
Yeah, not really. I mean, so for example, we know that with C9 ALS, the C9 mutation and the pathology arising from it can affect other cell types than just motor neurons. So there is, the frontotemporal dementia. There are some cases of Parkinsonism with C9. So I think a lot depends on two things. It depends on the mutation itself and whether or not it has a propensity to have an adverse impact outside the motor neuron, which could be for many different reasons. The other is that some families may have combinations of other genes which set the stage or change the threshold for a disease. So a paper came out, this is now getting probably way too far into the weeds, but an amazing paper came out in Nature, premier biology journal in Thanksgiving a year ago, showing a large family in South America where people were ravaged by having mutations in a gene called presenilin-1 that causes Alzheimer's disease. And then they found one woman in the family who had the mutation and did not get Alzheimer's disease. And they identified in her two other gene mutations, which probably blunted or essentially defanged, ameliorated, attenuated the effect of the mutation. But that illustrates very clearly how, there's some fancy names for it, but basically it's multigenic impacts which can affect susceptibility.
So Dr. Harms showed us last week that ALS discovery timeline that you showed us earlier.
Yep, that's his slide. I had his name in the fine print in the bottom. Full disclosure, that is his slide.
That SOD1 is listed on there, but we've actually had several patients that have the SOD2 mutation, and I've never seen the SOD2 listed on any of those ALS discovery timelines, even though some of these people have ALS. Can you talk about that a little?
Yeah, I don't know of any data that SOD2 mutations cause ALS. In other words, people can have a SOD2 mutation, but that doesn't mean it caused the disease. I mean, I'll believe it because we look hard for it. When you show me a family where there are a bunch of different people, many generations, who all have ALS, and they all have the SOD2 mutation, then I'll believe it. But that kind of family hasn't been seen yet.
Okay, so if we have people that have SOD2, we should have them get a hold of you.
Yeah, I mean, the question is, does it run in a family? I mean, we looked very hard for that back in the early 90s, and we couldn't find it. And the field in general has not found that. So I just don't know of data that the SOD2 causes ALS. Okay. Yes, people might have mutations, but God knows how many genes I have mutations in, or you or somebody else. That doesn't mean they cause a specific disease.
Okay.
Good. It's a good question. It's a very interesting topic.
What is the role of genes related to glutamate or GABA in ALS?
Yeah, well, there is a strong argument that part of the pathology in the disease has to do with excessive firing of motor neurons and the energetic demands that imposes. And to the extent that different genes that affect levels of glutamate affect the firing rate, they can potentially be important in this disease. It is interesting that when you look at that list, to go back to what Michelle said, when you look at the major ALS genes, by and large, they're not genes that affect glutamate. So the genetics does not lead you by the nose to glutamate. Right. But nonetheless, to try to answer your question, I can easily imagine that genes that modulate the activity, the tone, the firing frequency related to glutamate might have some impact on either the severity or rate of progression of the disease. I don't really have a good answer to your question.
That's fine. I appreciate it.
The next question, any suggestions to help families with mutations find other families with the mutations of the same gene or the same variant?
Yeah. Well, I think a couple of things to say. I mean, there are organizations at the NIH, you probably know a lot about them, called like NORD, National Orphan Disease, which look for orphan diseases, and ALS is one, and try to assemble all those mutations, and then try to find ways to co-relate them or interrelate them. And so NORD might be one way to do it. I think, your organizations like yours could play a huge role in casting the net to look for people. I mean, we think it's incredibly important to enroll all the folks in these registries, with or without familial ALS, but certainly with, and then within the registries in an anonymized manner, of course, to indicate which mutations are involved, with essentially a directorate, which is you, who could then, when the right circumstances arose, relate individuals to one another. That has to be done carefully because of HIPAA, as you know, and protection of privacy. But I think a group like yours could have a big role in that. And it ought to be done nationally. I mean, it's a big deal. It's a very good question.
We've started collecting some data like that, and specifically locations of people on this call specifically, and within our community too. So we definitely have already talked about and started the process of centralizing that data within a patient advocacy group so that we can cross different systems and use it maybe a little bit more widely. So,
I mean, to be honest, I would strongly suggest going to the ALS Association and saying, look, this needs to be done. We're doing, can we work together? To register that is not a high cost expense, but the information is very, very valuable. And I don't know, but I'm guessing, I mean, they have a network, you have a network. It's ridiculous that these networks are not, at least for some projects, working together.
absolutely.
So Dr. Brown, we do have a deal with MGH is giving us a free software platform for all of their patient registry. And so we're in the middle of getting the software deal done so that we can actually host it. So we're working together. So MGH has been extremely helpful.
Yeah, and I would just say that, there's new leadership at ALSA. First of all, their track record in helping ALS is really spectacular, but there's new leadership which is very receptive to new creative ways to help solve this problem. So I wouldn't be shy about reaching out to them.
Yeah, thank you.
Yeah, I'm glad you're doing
that. As I'm sure you're aware, genes that disproportionately impact Hispanics and African Americans, do you know of any studies that may explore possible familial mutations in those populations?
Yeah, so that is a super important issue. It is not well studied. It begs study, it begs scholarship, and that's another reason why we need these registries. Okay, so, there have been very few studies that looked, for example, at the incidence of these genes in Africans. That's starting to happen, or in Black Americans. So that is a hugely important, or Latin Americans. I mean, the genome databases now are beginning to be large enough to give you a few hundred or even a few thousand individuals from different geographic locations. So what you're asking about, I think, will increasingly be possible. But we've talked about it many, many times at meetings, and just hasn't been done. I mean, Africa's really incredibly understudied in terms of so many of these things, and yet it's there. I mean, what can you say? So very, very important topic. Again, no one agency can do everything, but it's the kind of thing with the right repository where you could make a big difference.
Wonderful. So now we're going to get into a few NurOwn questions, and I know a lot of the audience is chomping at the bit to hear some of your answers. So we start by telling you we know that you are PI and that you can't release some data, and that you have to be circumspect about that. But we also know that the trial hasn't been unblinded to the PIs and patients as to who was on NurOwn and who wasn't. So just in your 40 years or so of experience, did you observe any treatment effect in any of the patients that you had that were participating in the trial?
Well, I mean, there are one or two patients who are sort of legendary, and I won't call them out by name now, but I see at least one of them smiling. And so there are some exceptional number of folks who've had a very dramatic response. The problem is that representative of the group as a whole? That's the question.
Sure.
And when all the data are broken down, we'll know the answer. I do think, I'll repeat, I think there is a benefit from the stem cell therapy. The question is, when do you have to give it? How big is the benefit? And what does it mean in terms of translating into widespread use? I think they have a benefit. I have several reasons for wanting to say that.
So you kind of started to answer the next question, talking about how could NurOwn modify neurodegeneration, neuroinflammation, and neuroprotection, and yet not have relevant clinical effect. So how does that make sense for our audience to understand?
Well, the question is it adequate intervention in each of those realms to make it the overall clinical course is in some way the integration, the composite of each of these events that are adverse and are occurring simultaneously. And the question is, which is most important, and how big is the effect of any intervention, not just stem cells, on each of those components? And what do we really need to do to get the Achilles heel of the disease?
Okay. So our next question actually comes from feedback that some of the patients are getting from their neurologist. And so obviously you and Merritt and a few of the other leading PIs have more insight. So some of the people have said, we think NurOwn is going to need a second double-blind placebo trial to follow up on this. And so given what you've seen, do you think it'd be ethical to do another double-blind placebo when we know that there's some people that have already been responders?
Yeah, well, I mean, the question is, if the company feels it wants to shore up the results in some way that defines how best to give it and who best to receive it, in theory, that might require another study. Now, the question of how long you can do a placebo for is a big challenge. To ask people to go a year without being treated is really a huge ask. And, I have trouble as a clinician with that, and I'm not sure I would participate in a study again where there was a placebo trial that long. Most of the new studies, and all this, are like 6 months placebo. And most of them guarantee drug at the end, no matter what. So there are some ways to think about making it more plausible to do more trials. But a year of placebo is a big, big, big ask. That's a huge thing to ask of anyone.
I think the community agrees with you on that. So, Sarah?
Absolutely. This kind of goes back to the people in the NurOwn trial. I know there were some that had ALSFRS scores that were 25 or below. And we know that this might have convoluted the data a bit. How does this make it difficult for approval?
Well, you basically said exactly the situation. The question is, what population of people respond, and how good is that response? And is it good enough to justify general approval or only approval for that particular group that responded? These are great questions, and they're pretty generic. And I certainly don't have the answers to them. Radicava is an interesting talking point because it came, first of all, from outside the US. Secondly, not everyone responded to Radicava. It was a subset of folks, and yet they got approval for use across the board. And I would think it's very, very hard to get an ALS drug approved just for a subset of ALS patients. It's one thing if it's a specific mutation. Okay, that's one thing. But if you're going to take all, say, sporadic ALS and only treat people who are at a certain stage of the illness and not others, it seems to me that's a very fine hair to split. That'd be very hard to do, I would think. My own view is it ought to be all or none. But if it takes more study to understand exactly how best to use it, the company will adjudicate that on its own.
Can you explain the mechanism of action of copper ATSM on SOD1?
Not very well. So that's been put on the map by a very brilliant chemist named Joe Beckman. He's done a wonderful job with it. And basically, it's modifying copper toxicity by binding copper is the simplest explanation I can come up with. And I think that's probably a fair statement. Copper is very, very toxic. one of the major binding substances for copper is SOD. And I think what the ATSM does is modify buffering of copper in a way that's beneficial.
This question comes from the chat. It's kind of talking about biomarkers. I know we are currently doing research into speech and voice as a biomarker. And the question is, how long do you think until ALS has a significant enough biomarker that A, for diagnostics, and B, for maybe placebos in clinical trials?
Oh, I think they're there now. I think there's some very good biomarkers. You know, the ones that I'm sure you've heard the most about are markers of neurofilament breakdown, you know, so-called neurofilament heavy chain, light chain. Some of those are just almost as good in the blood as in spinal fluid. So I think they're there. Now, we probably need more. Those are markers of breakdown of neurons. There are other markers which tell you how much inflammation there is. And so we probably need those as well. And there may yet be other types we need, but I think you will find most of the studies that are being done now do incorporate the degenerative, that is to say, the neurofilament biomarkers. I don't know if that answers your question, but I mean, as we all know, the real promise of biomarkers is that if they're really good, they may show you benefit long before a clinical trial shows you benefit. Wouldn't it be nice if you could do a study in 6 weeks? Forget a year of placebo, just say 6 weeks, 2 months. And that may happen. That may happen.
And that is one of our focus. We're trying to focus on digital biomarkers.
Yeah,
so we're trying to bring speech and different wearables for biomarkers.
Yeah, that's wonderful. That's fantastic.
We want to say thank you so much, Dr. Brown, for joining us tonight. This has been very informative and has helped make understanding of kind of where the genetic therapies are going in the future. I know I got a lot of questions answered myself, and going to probably have to watch it again. So to be able to fully understand and grasp everything that you talked about tonight. So I want to say thank you.
It's a great pleasure. As I scan the Zoom, I see many friends, and I'm so honored to be able to speak with you. You know who you are, and I'm smiling at you. I don't know if you can see that, but I'm so glad to be able to participate in this. And I thank you folks all for what your group is doing. I think it's just terrific and badly needed, and it'll make a huge difference. So thank you very much. Thank you.